scientist Pam Akinyemi. We are going to play a game where we test each other's knowledge over topics that affect stress responses in differing species.
So Lauren, let's start off with a simple question. What is stress?
Well, stress is a change in the cellular composition of an organism in response to abiotic factors like temperature, pH, pollutants, and biotic factors such as species. There are different types of interactions that can cause stress. Pam, can you explain this?
Well, there are three main interactions that have been tested to show stress. The first is a predator to prey, where the predator benefits from the prey, for example, mice and snakes. The second relationship is a parasite to host, where the parasite benefits and the host is harmed or unharmed. An example of this would be fleas on dogs, as the relationship is harming the dog and causing unneeded stress. And finally, the last relationship is competition between two species. This type of relationship will be our main focus in today's episode. This relationship harms both organisms involved and causes equal or differing levels of stress as they both fight for survival in a limited niche space.
Now let's delve into what specific types of responses we see in the presence of a competition. This episode we'll be talking about a specific experiment carried out by Alex Gunderson and colleagues that tested the cellular stress response of competing crab populations under global change.
Hi, I'm Danielle, another scientist on What's Going On. So I'm sure you're all wondering how exactly the study was carried out. First of all, there were two types of crabs, the P. cinctypes and the P. maninaculus. The crabs were taken from California and stored in the lab. In the lab, they were put in flow through sea tables that were maintained at 13 degrees Celsius. There were three different environments: a heterospecific one which had individuals of different species, a conspecific one with individuals of the same species, and lastly, we had a control environment that only included the focal crab. They were going to quantify competition by counting the number of interactions involving a focal crab. This focal crab would be tagged with paint so it would be recognized. To quantify competition, they counted the number of interactions the focal crab participated in over a 1 hour period. The observer counted all the interactions in a dark room under a red light to limit the disruption of behavior. At the end of the trial, the focal crab was frozen and ground to powder, and the RNA was extracted from the powder. And then the RNA was assessed.
Hello, I'm Riley from What's Going On. I'm going to walk you through the results of this study. Vectors principal component 1 and principal component 2 are uncorrelated orthogonal representations of our variables. In principal component analysis, principal component 1 has the most variance and therefore can account for the most variability within the experiment. In this experiment, principal component 1 is a linear combination of these gene expressions. As you can see, principal component 1 gene expression is unrelated to the number of agonistic interactions with an ANOVA test p-value of 0.944.
Welcome back. We hope you all enjoyed the experiment of the week. Now let's discuss what exactly these results mean. Pam, take it away.
Here, figure 1 shows that the porcelain crabs do engage in agonistic interactions with one another, but more specifically, individuals experience more agonistic interactions with heterospecifics than conspecifics.
Figure 2 shows that there is no evidence to suggest that interactions in a short time frame can lead to increased cellular stress response levels in a species. In addition, there was no difference in gene profiles between the focal individuals who interacted with heterospecifics and conspecifics.
Figure 3 shows that there was no correlation between the number of agonistic interactions and gene expression, PC1. Because these results varied with the results of previous experiments, we realized that next time we may need to extend the time of interaction. Also, the interactions may not have been aggressive enough to cause a stress response.
So what have we learned today? Let's go over the basics. Species interactions can result in an induced cellular stress response, which can influence a species population, community-level processes, and consequences of anthropogenic global change. There are three types of interactions that we can study, and they are the same type of species interactions that we've all studied in class. Host to parasite, predator to prey, and lastly, competitive interaction. The takeaway from today is to understand the consequences of species-induced and abiotic stress under anthropogenic global change.
And that's what's going on.